To understand the physical reality of biological inheritance, we must scale down into the nucleus. Here, on paired homologous chromosomes, the instructions are written. An allele is an alternative form of a gene. It occupies the exact same physical location, or locus, on a homologous chromosome as its counterpart, but it differs in its specific sequence of nucleotides. This definition evolved from abstract beginnings. Between 1856 and 1865, Gregor Mendel observed traits like seed color in garden peas existed in predictable, contrasting forms. He tracked heritable factors segregating into offspring, though the molecular mechanics remained invisible to him. It took the later work of Thomas Hunt Morgan and his colleagues at Columbia University to anchor these abstract traits to physical space. By mapping sex-linked inheritance in fruit flies, Morgan proved alleles occupy specific fixed positions on chromosomes. A mechanistic definition arrived in the mid-20th century with the discovery of the DNA double helix. This structural revelation allowed geneticists to define an allele explicitly as an alternative base pair sequence. Every allele is a tangible string of biochemical data. Variations in that sequence physically alter the biological output of the organism. This diagram outlines a structural taxonomy to categorize that biochemical diversity. We can organize every known variant across 4 distinct branches: dominance, function, origin, and population dynamics. The phenotypic consequence of any given allele depends on exactly where the sequence variation occurs, whether a single nucleotide polymorphism, a sequence insertion, or a large copy number variant. Location determines impact. If a variation occurs within the coding sequence, it alters the amino acid structure of the resulting protein. If that same variation strikes a regulatory region, like a promoter, the protein structure remains intact, but the cell might produce too much or too little of it. This functional architecture provides the framework for the genotype-phenotype correlations we will map across our hierarchy. Exploring the dominance branch requires stepping away from the classical idea of one trait simply masking another. At the molecular level, dominance is a matter of protein dosage. This chart demonstrates wild-type haplosufficiency. Here, a single healthy allele produces 50 units of protein. For this specific gene, 50 units is enough to cross the biological threshold and sustain normal cellular function. If a mutation disables the second allele, its output drops to 0. But because the first allele still produces 50 units, the cell survives. In this state, the loss of function mutation behaves recessively. The remaining allele compensates for the deficit. But for some genes, like the tumor suppressor RB1, the survival threshold is higher. If the cell requires 80 units to function, the 50 units from a single wild-type allele fall critically short. This is haploinsufficiency. The loss of a single copy causes the cell to fail, making the mutant allele dominant. Biological minimums, the specific protein volumes a cell needs to survive, dictate whether a mutation behaves as dominant or recessive. Sometimes, different alleles operate with equal priority in the cell. In codominance, both alleles are fully and simultaneously expressed without blending. In the human ABO blood group system, possessing both the A and B alleles results in red blood cells that display both specific surface antigens. This is distinct from incomplete dominance. Here, a single wild-type allele cannot generate enough protein to create the full phenotype, resulting in a blended intermediate. Crossing a red and white flower to produce pink offspring occurs because the total pigment dosage is cut in half. Furthermore, an allele's expression can depend directly on its environment. Conditional alleles produce mutant phenotypes exclusively when triggered by specific external factors, such as shifting above or below a critical temperature. These temperature-sensitive mutations are highly effective lab tools. Geneticist Leland Hartwell utilized conditional alleles in yeast to switch essential regulatory genes on and off using heat. This targeted approach isolated the primary mechanisms of cell cycle checkpoints. An allele is a responsive biological unit. Its final expression is sensitive to its allelic partner and the precise environmental context in which it operates. Moving to the functional status branch of our graph, we use the classification system developed by Hermann Joseph Muller in 1932 to measure exactly how mutations scale biological output. At the very bottom of the scale is an amorphic or null allele. A structural disruption, like a frame shift or nonsense mutation, forces the ribosome to detach early. The sequence generates 0 functional product. A hypomorphic allele is slightly more productive. Often called a leaky mutation, a minor sequence error allows translation to finish, but produces a protein with a deformed structure and reduced activity compared to the wild type. Conversely, a hypermorphic allele generates excessive activity. By duplicating a promoter region, RNA polymerases swarm the DNA sequence, resulting in a dominant gain of function where the cell is flooded with normal product. Amorphic, hypomorphic, and hypermorphic alleles form a clear sliding scale, quantifying direct increases or decreases in regular genetic dosage.
Beyond simple dosage adjustments, some mutations introduce new biological instructions. Neomorphic alleles are dominant mutations that generate a novel function or expression pattern that is absent in the wild type. The antennapedia mutation in Drosophila is a classic neomorph. A chromosomal inversion places a homeodomain gene under the control of the wrong promoter, instructing the fly to sprout legs directly out of its head in place of antennae. Even more destructive are antimorphic or dominant negative alleles. Instead of simply failing to work, the product of an antimorphic allele actively antagonizes and disables the healthy products generated by the wild-type allele. We see this mechanism driving human cancers via the TP53 gene. The healthy p53 tumor suppressor protein functions as a 4-part tetramer. When an individual inherits one antimorphic allele, a deformed mutant subunit is assembled into the structure alongside 3 healthy pieces. That single deformed inclusion destabilizes the entire complex. Even though the cell possesses a healthy allele producing normal subunits, the mutant product binds to them and drags their collective functionality down to 0. Antimorphs are uniquely dangerous because they actively weaponize the cell's own protein assembly machinery against it. While a single diploid individual carries at most 2 alleles for a given autosomal locus, scaling up to the population level reveals immense diversity. A freely interbreeding population can maintain thousands of distinct variations of a single gene, known as multiple allelism. The major histocompatibility complex, or MHC, in vertebrates contains over 6,000 known alleles at a single locus. This staggering polymorphism ensures the population can identify and present an incredibly diverse array of pathogen-derived peptides to the immune system. The precise immune surveillance mechanisms relying on this diversity were detailed by scientists including Rolf Zinkernagel, an achievement recognized by the Nobel Prize in 1996. Tracking down the origin branch of our taxonomy reveals that sequence variation isn't the only factor dictating expression. Through genomic imprinting, an allele's expression depends on whether it was inherited from the biological mother or father. Consider the UBE3A gene in human neurons. Both the maternal and paternal alleles share the same sequence, yet under normal conditions, only the maternally derived allele is actively transcribed into functional RNA. The paternal allele remains silent due to targeted regulatory interference. An antisense RNA transcript, reading in the opposite direction, is expressed from the paternal chromosome. It physically collides with the transcription machinery attempting to read the UBE3A gene, stopping it in its tracks. Through epigenetic methylation and histone modification, these parent of origin marks supersede the nucleotide sequence, dictating functional outcomes based on inherited origin. With so many possible mutations, geneticists rely on operational tests to determine if 2 distinct recessive mutations occupy the same gene or 2 different genes. The definitive tool is the cis-trans, or complementation test. Place 2 mutations in a trans configuration within a single doubly heterozygous organism and observe the phenotype. If the mutations reside in the same functional zone, or cistron, neither allele provides a working product. The alleles fail to complement. However, if the mutations exist in different genes, they successfully complement each other. Each chromosome provides a healthy product for the other's deficient gene, and the organism appears wild-type. There is a rare exception, interallelic complementation. Here, 2 distinct mutant alleles of the same gene encode damaged protein subunits that, when combined, stabilize each other and restore partial activity to a multimeric complex.
Despite that exception, the complementation test remains the genetic criterion for definitively mapping the boundaries of a genetic locus. Once alleles are mapped and categorized, we track them across generations. In population genetics, allele frequencies constantly shift due to genetic drift, mutation, and the pressures of natural selection. Often, directional selection drives a favored allele toward total fixation, but balancing selection maintains multiple alleles at intermediate frequencies. A primary mechanism is overdominance, or heterozygote advantage, demonstrated by the sickle cell trait. A single nucleotide substitution in the human beta-globin gene creates the HBS allele. Homozygotes possessing 2 copies of this allele suffer from severe hemolytic anemia as their red blood cells distort and collapse, yet the allele persists in specific geographic regions because heterozygotes, possessing just 1 copy, gain resistance to severe malaria. The evolutionary forces acting on these frequencies are measured against a stable mathematical baseline, the Hardy-Weinberg equilibrium, which predicts how alleles distribute into genotypes in the absence of outside pressure. From predicting population shifts to tracking individual amino acid substitutions, every facet of inheritance connects back to this central hierarchy. Alleles are the physical units of biological variation. They dictate the folding of a single protein within a cell, and by extension, they drive the evolutionary trajectory of our entire species.